Hi everyone. It's lovely to be invited along to this event this evening. Myself and my colleague Charlotte, we work in the Newcastle mitochondrial lab, and we're going to talk to you a little bit about how we make a diagnosis of mitochondrial disease in the laboratory. I didn't think my slides were going to move there, but they did. So just to give you a bit of an outline of what we're going to talk about, in this presentation, we'll explain why a clinician might think an individual has mitochondrial disease or mito, and how we would test for this in a lab. We'll then discuss what happens if we don't make a diagnosis and why it's important that we keep improving the lab tests that we're able to offer in the NHS to ensure that we help more families reach a diagnosis in the future. And then we'll hand over to Will and Renata from the London team, who will talk a little bit about the Lilly Precision Medicine study and the tests being performed as part of that study. So just a very busy slide, but I'm not going to go through this in great detail, but I'm sure as most of you are aware, mitochondrial disease is genetic. It results from changes in our DNA, which we call variants. And mitochondria actually contain two different types of DNA. They have nuclear DNA, and we inherit one copy of each gene from each of our parents, and mitochondrial DNA, which we only inherit from our mothers. Nuclear DNA, it's huge. It contains more than 20,000 genes, and those genes are used to make proteins that the body requires. And of these 20,000 genes, more than 1,000 of them are thought to be important for the functioning of mitochondria. And so far, more than 400 have been associated with mitochondrial disease. Mitochondrial DNA, on the other hand, it's tiny in comparison, and it contains only 37 genes. So as you will know, the main function of mitochondria is to make energy required by the cells in our body to function correctly. However, mitochondria have many other functions too, and all of these functions of mitochondria can be affected by mitochondrial disease. Mitochondrial disease can have many different clinical symptoms affecting many different parts of the body, and this can make mitochondrial disease quite difficult to diagnose in the clinic. But often when there are multiple different body systems involved, such as heart, eyes, brain, muscles, then doctors might suspect mitochondrial disease. So just a quick view of how genetic testing is organised in the NHS, and I won't go into this in any great detail, but back in 2018, all genetic tests was sort of reorganised into 7 genomic laboratory hubs. And what this means is that we offer a defined set of genetic tests. It ensures that no matter where an individual lives or in which hospital they're seen, the same genetic test can be ordered. And 3 of these genomic laboratory hubs work together to deliver the rare mitochondrial disorders service. So the 3 labs are in London, Newcastle, and Oxford, and they're linked to specialist clinical teams in each city. And the labs work together to ensure that we deliver the same standard of genetic testing across the national service. So we thought it might be helpful to explain kind of what happens to your samples once they've been collected in clinic, because I would imagine your sample goes from clinic and then you're waiting for the results, and this can probably seem quite a long time to be waiting. So if a doctor suspects this could be mito, samples are taken, usually blood in the first instance now, and that's sent to the lab for testing. Once we get the samples in the lab, we book them into our lab computer systems, we label them, and we store them correctly. The referring doctor will have completed some paperwork with a brief clinical or maybe extensive clinical history. And this is used to determine what is the most appropriate test to be assigned. And that's done on a case-by-case basis. We'll also consider the patient's age and the sample type we've received. It's really important that we assign the correct test so we make sure that everybody has the best chance of reaching a diagnosis. We couldn't list every sample for every test. It wouldn't be needed, and it would really quickly overwhelm the lab's capacity to do the testing. And what that would mean is that it would take far too long for anybody to get any results back. So what we really need is the correct test for each individual. So once the samples are booked in, we've assigned the testing, we extract the DNA from the sample, and we use it sounds fancy, but we use a DNA extraction robot or machine in the lab. Once the DNA is extracted, we can perform the tests and analyze the results. The data or the results can be in many different formats depending on the tests we've performed. And then we write a report explaining the result and we send this back to the doctor. The results are then discussed at clinic with the individual. And just to highlight, where possible, we do store the remaining sample in the lab so that if in the future more tests are required, hopefully we can do that without having to collect more samples. So I think it might be helpful just to talk a little bit about some of the tests that we can do and the ways we look for mitochondrial disease, and also explain how it can take sometimes what must feel like quite a bit of time to get these results back. So as I mentioned earlier, we extract the DNA and we look at nuclear DNA or mitochondrial DNA or both. And when we look at nuclear DNA, we can look at a single gene of interest, a group of genes, which might be a small number of genes, maybe 5, 10, or sometimes it's hundreds of genes altogether. Or we can look at a whole exome or a whole genome, and I'll just explain what I mean by that. So genes are made up of areas of code that are responsible for making proteins. And these are called exons. And they're just shown in this picture here. I don't know if you can see my pointer, but the light blue bars are what we call the exons. And the bit in between, these grey parts, are non-coding and they're called introns. And normally, well, not normally, but often mitochondrial disease would be caused by a change in the exon. So when we look at a single gene or a group of genes, we normally just look at the coding exons. When we perform exon sequencing, we can look at all exons of all genes. So we're looking at far more genes, but still just looking at these exon sequences. When we perform whole genome sequencing, we're actually able to look at all exons and all introns, or at least we get the data for all exons, all introns, for all genes. So we get a huge amount of data back. And what we need to do is we need to have a look at the data that we receive back. We need to look for changes in the DNA that is different to what we would expect to see. So most of the changes, we'll see hundreds of changes, and most of these changes do not cause mitochondrial disease. And what we have to be able to do is identify the changes that do cause mitochondrial disease. And that's the job of the lab scientist to do that. This bottom image it's a complex image, but it's just some data that we receive from mitochondrial DNA. I wouldn't worry too much about all the grey parts, but if you can see the coloured lines running down, this just shows where we've seen different changes in the mitochondrial DNA. And these are different variants that we need to investigate. So I think when we're looking at a variant, we have to work out whether it's important or not. And how do we do that? So we have national and international guidelines to help us do this. And it's important that we follow these, because what it means is that if a variant is seen in one lab and in another lab, then they are scored in the same way across different labs, and we'll come to the same conclusion. So it's a sort of points-based system. I think the first thing we would look at is how common is the variant or the change that we've seen. If it's common and it's seen in many individuals, this can be discounted. These common variants are what we would call benign or lightly benign, and they do not cause mitochondrial disease. If a variant is rare, then it's more likely to cause disease, but not guaranteed by any stretch. So then we have to think about what gene has the variant occurred in? What's the function of that gene? Could it fit with the presentation of the individual? And also look about where it occurs in that gene, because some parts of the gene are more important than other parts of the gene. So there's a lot we have to do. And we would also have a look and see, has it been reported before? Has anybody seen it in any other families? And all of these bits of evidence are given a point or multiple points, and we sort of add them up. So if they get a high score, a 6 or above, they're pathogenic or likely pathogenic, and we are certain that they cause mitochondrial disease and the clinical symptoms seen in the individual. Other variants might not have so much evidence, but can't be excluded from our analysis. And these are called variants of uncertain significance, or VUSs. Sometimes that's how they're described. And in this case, we haven't got enough evidence to say that it causes disease, but we also aren't sure that it doesn't cause disease. So we need to investigate these further. So to recap, what we would do is extract the DNA, usually from blood, look at individual genes, groups of genes, or all the genes, and then interpret the changes in the DNA that we've identified. And from this, we can either confirm a diagnosis, identify a variant of uncertain significance that needs further investigation, or in many cases, we won't make a diagnosis. And then these variants of uncertain significance, or if we haven't found a diagnosis, this is when we need to do further work. And Charlotte, my colleague, is going to chat to you in just a second about the different types of work that we can do. It's just useful to note that all the lab tests have a set turnaround time. So this is a time in which from getting the sample, we need to report the results. And these are nationally defined and everybody needs to work to these. So for genetic testing, they range from 3 to 42 days. And that's dependent on how urgent the test is and how complex the test is. So for example, a prenatal test, so a test done in pregnancy where we need to inform a pregnancy, will be done in 3 days maximum. A complex test of hundreds of genes, we're given 42 days to do that. And that's just because the length of time it takes to get the data, but mostly to do all this interpretation of all these different variants that we find and figure out if we have got a diagnosis in there. So if I can now hand over to my colleague Charlotte, and she'll explain about about the additional lab testing that can be performed, how we can make a diagnosis, and also about the introduction of new technologies that will help improve diagnosis rates. Charlotte, over to you.
Right. So thank you, Emma. So yeah, I'm just going to talk a little bit more about the extra tests that we can do to help prove that a gene variant that we find is actually causing the symptoms that the patients have. So, and some people on the call, they may have had a muscle biopsy or a skin biopsy, because that's often how we used to test for mitochondrial disorders. And now we do the genetics first, and then we do these functional tests as a sort of second line test to see if we can prove that they are responsible. So the first two tests are the histochemistry and the biochemistry. And these are accredited, which means that we're sort of licensed to do these. We have people that come in and test our methods, that test our knowledge, our safety to practice, to ensure that we're doing everything sort of by the book. So the histochemistry and the biochemistry uses either muscle biopsies or skin biopsies to look at the individual parts of the mitochondria to see how they're functioning. So for biochemistry, we give the mitochondria all of the things that they need to do each individual part of the energy production, and then we monitor its ability to do it. So we look at each of the different parts in turn, and then that helps us get some evidence. So if it might be that the gene problem that we've identified, or we think we've identified, is in a particular type of mitochondrial protein. So if it involves different complexes, for example, we would want to see that we've got some, it's not performing as well as it might do in the controls, which would be the red panels there. So Lang Ping is there, the spectrophotometer doing the biochemistry in the panel underneath. The histochemistry that we do, again, it's looking at mitochondrial protein levels and how well the mitochondrial proteins are, whether they're present. So the blue bits in this panel at the top, so the bottom right panel, the blue fibers that we can see, each of those sort of individual blocks is a muscle fiber. And so we're looking there to see how the mitochondria are functioning. So that's a complex 4 deficient fiber is the blue one. And that's an evidence of that the mitochondria aren't performing as well as they might do. We don't just see these blue fibers in people that have mitochondrial disorders, and they also happen as we age normally. So it's important to consider all sorts of things besides the variant, and actually putting it into the sort of position of the patient and the clinical symptoms that we have to try and determine if these are important changes. And that's Sela at the bottom. So Sela is at the, she's essentially cutting sections of frozen muscle using it's called a cryostat. The muscle biopsy sections that we use for these stains are, if you had 8 of them stacked on top of each other, it would be the width of a sheet of paper. So they are incredibly thin slices of muscle that we use for these stains. But the muscle biopsy is really important for some of these tests to get this evidence. On the right-hand side, I've just shown a couple of examples of functional tests that we can do. These aren't accredited. They're performed on a research basis at the moment. And we have people who are specially trained to do this. And for these, we're looking at individual subunits or parts of the mitochondria to see if they are present. So on the top line, hopefully for underneath P1 and P2, which stand for patient 1 and patient 2, the top blocks are missing those black lines. So that each black line corresponds to a particular part of the mitochondria, a particular protein that's involved. So you can see for CO1 and CO2, which are our controls, they're people that do not have a mitochondrial symptom. They have plenty of black showing in those lines, whereas for the patients, they do not. So if we can do this for individual proteins, which is on the left, or we can look at groups of proteins, which is on the right. So each test gives us a bit more information or slightly different information to try and put together this picture of whether or not the variants that we found are causing the mitochondrial symptoms that we have. Because if we're doing a genetic test, we may find, if we do an exome, for example, there's thousands of changes that make us all different from each other. They don't necessarily cause any kind of presentation or any kind of clinical symptoms, but obviously there are some that do, and we really have to sort of filter through the haystack to find the needle, because that's the kind of situation that we're facing sometimes. And we need to give everybody the right answer as well. So it's really important that we build a strong case and we assign the correct codes and try and get as many points as we can to make sure that we're getting the right answer. Can I have the next slide, please, Emma? Thank you. So the NHS approach to diagnosing mitochondrial disease, I like to think of it a bit like the sort of the iceberg, because the NHS is stretched. You hear about this in the news. We have a limited pot of money. We have limited staffing. We need to make sure that we're providing value for money, and we need to get it right. The testing that we do has to fit into the reporting times that we have. So we need to make sure that we can do it for everybody in the timescales that we say we can. So that means that we're sometimes looking at what can seem like the tip of the iceberg. Compared to where we were 10 years ago, we are looking at so much more now in terms of the number of genes than where we were then, because the technology's improved. So it means that we're offering a better service with the resources that we have. When you think about the iceberg, there's all of the iceberg underneath the surface that we don't see. So the top bit is looking for known variants in known disease genes, but then there's an awful lot of other different classes of variants and genes that we can't look at within the NHS at the moment, because we just don't have the sort of the capacity to do that and to do it well within the time that we have. So then looking down the iceberg, there's sort of different tiers of difficulty. And what we want to do really is to lift up the iceberg so that instead of it being an iceberg, it's a mountain. So eventually, with improvements in technology or the way that we're doing things or whatever we can get our hands on to make things better, we're moving up so that we can then identify new technologies to help us work out variants of uncertain significance that we know are probably likely to cause disease, but we don't have enough points there to show that it definitely does. Then looking down the next spectrum where we've got the new disease genes that haven't yet been known to cause mitochondrial disease. And then the ones underneath that, which are the novel genes full stop, that we don't necessarily think are mitochondrial, but actually somehow are interacting with mitochondria. And that's the really sort of challenging part, and trying to get everybody the diagnosis when they might fit in different scales of that iceberg and different levels is quite a challenge. But we are here for the challenge. We don't do it because it's easy. We do it because we're committed to doing it and doing a good job. Next slide, please, Emma. So from the research side of things, we are working on all levels of that iceberg. So not just the patients that get the diagnosis straight away. We are working to try and turn those variants of uncertain significance into a diagnosis. Some of the things that we do are by reporting when we find new disease genes. So these are new genes that have been identified that have got spelling mistakes in them by our team and the London team. To just show that, we are trying to get the message out there, because then once we've proven and we've done all the hard work to say, look, this is a new cause of a mitochondrial disorder, we can then implement that within the diagnostic pipeline. So instead of it being something at the bottom of the iceberg, it then moves it up to the top. And then we start looking for these in our routine diagnostic testing. So every patient then can find the variants that would have been in the genes at the bottom. So we're trying to sort of evolve our testing in any way that we can. And Panel App is the way that we do that. I'm just tricking Emma because she thought I was going to move on then. So the Panel App is the tool that we use in the NHS, and actually a lot of other international countries use this. So it's a really well curated list of genes that we know can cause all sorts of different disorders. So it's not just mitochondrial disorders, it's all rare diseases and cancer associated conditions as well. So it's the sort of, our goal is to report it in the literature, in scientific papers, to then say to Genomics England and Panel App, look, we've got a new gene that we need to add for testing. And that's how then that increases our chances of getting a diagnosis. Next slide, please. So the other way that we're doing this is sort of moving away from sort of the NHS diagnostics, but into the research lab to then bring in new technologies. So this slide just shows two different mechanisms. The LifeArc Centre, which is partnered with Lilly for rare mitochondrial diseases, is looking at how we can take new technologies and bring those into diagnostics. So the example that I've shown here is for proteomics, which is looking at proteins instead of DNA. So proteins are like the end point of the DNA. They're the bits that do the work in the mitochondria. And so we can look at all of the proteins to say, where are the problems in the mitochondria, we've got more proteins perhaps, or which ones have less, and different things can cause different problems. So that is also not easy to do, but it's a different sort of bow to our armor. And hopefully we'll be able to get some diagnoses using hopefully blood going forwards, and this can be done quite quickly. So bringing that into diagnostics is one area where I think we're going to see a massive spike in the diagnostic ability that we have. And also the Network of Excellence, which is a sort of, it's NHS funded, is a way of introducing new technologies within the diagnostic lab as well. And I think we're going to pass over to Renata and Will now, because really they're going to talk about the next steps for rare disease and how can we do this? How can we do it better? How can we do it faster? And how can we do it cheaper? Because as much as we ask for more pennies from the government, they don't seem very keen on finding us any. So we're trying to be a bit canny with what we do to get more bang for our buck, really. So it's how we can make things better with the resources that we have. And I think genomics, together with proteomics and transcriptomics and the technologies that Will and Renata are going to talk about, are going to be really powerful in years to come. And I think that's me. Thank you.
Can you now see my slides?
Looks great. Thank
Great.
you, Renata.
So, Will, do you want to start with?
Yeah, sure. Thanks. So, hi everyone. I'm Will. I'm one of the genetics doctors down in London, and I work in the adult clinic in Queen Square, and then in general genetics in Great Ormond Street. And Renata, who's just on the call there, is our senior bioinformatician. And Rob, one of the lead doctors in our services, on the call as well for the questions later. So just kind of following on from what Emma and Charlotte were talking about, we just wanted to expand this about what we can do to push further to find a diagnosis. So I'll briefly mention a few things about reanalyzing data, which will hopefully develop the concepts that Charlotte was just talking about there with this horrible iceberg. And then Renata is going to talk about two of these newer technologies. And then we just have one slide at the end talking about things that we might be able to do in the future in terms of using AI to get people a diagnosis a bit faster. So reanalysis then, why is it important? So I think what Charlotte really nicely laid out there was that just because we didn't find the diagnosis doesn't mean it's not there. Now, for some people, when you have a negative report and the likelihood that they had mitochondrial disease at the start was relatively low, then that is quite reassuring. But if someone really thinks you have mitochondrial disease and the test is negative, there might be something else to be found. So the first question is, are there new genes out there that were not known about during the NHS test? And this is why the point that Charlotte was making is really important about us trying to be doing some expanded work on the research side so we can find these new genes and feed them back into the system. So the next time the clinical scientists go to analyze a case, it will be part of the routine analysis. It could be that the variants of the specific genetic change or mutation, these spelling changes in the genes, it could have been missed for technical reasons. And technologies are changing all the time, and also the way we interpret them are changing. And so we want to, through the research that we do with our colleagues around the UK and also with the Lilly Foundation, to design systems to address these. So how can we do this? Well, one of the first things we do if we have a case where we're pretty sure they have mitochondrial disease, but we don't have an answer, is to look again at the clinical picture. So we could have said, okay, we've done lots of tests for mitochondrial disease, but when we go back over what's wrong with the patient, could it be that their muscle problem looks like mitochondrial disease, but is actually something else, or same with the brain problem or an eye problem? And so sometimes we have to go a bit broader with the things that we're looking at. The next thing then is sometimes we might need to say, okay, 3 years ago you had an analysis, but are there 10 new genes out there that we didn't know about at the time? So we try to keep re-updating these lists of genes that can be involved in mitochondrial disease. And this is hard to do at the moment in the NHS unless you have a big change in your clinical symptoms. It's hard to get the NHS to reanalyze things because the scientists are so busy. They're doing so much more work than they had to previously with basically no extra staff. So sometimes this kind of analysis is not impossible in the NHS. And then the other thing that we could think about is if we have more family members, could we look for variants or genetic changes that they share or don't share? So for example, if you have 3 or 4 people in a family with the disease, could we analyze all of them at the same time and the unaffected people and try to match mutations that are present in the affected people but aren't present in the other patients? And we call this type of analysis agnostic analysis because it's where we don't start with the list of genes that we're going to look at and we just let the variants pop out and then remove them if they're in the wrong people of the family. We especially do that where we have a mum, dad and a kid. And then the next one is thinking about, are there new programs? So bioinformatic tools. So when we say bioinformatics, that means that system that Emma was showing you. So we do all this sequencing, we read the sequence of the DNA, and then some very clever person on a computer like Renata has to design these programs to turn a jumble of letters into something that we can analyze. And so sometimes there's new tools being published there that we want to try out in the research setting, and then hopefully we'll be able to get it into the NHS. And then the last thing is these newer technologies. And so there's things, for example, long read sequencing, RNA sequencing, and proteomics that Charlotte just mentioned there. And in some cases, these can be really useful. So they're not a catch-all for everyone. It's not that because we did a normal technology to start off with and we do this that will diagnose everyone. Definitely not. But if we carefully pick the patients who are likely to be helped by these technologies, we can find new diagnoses. So Renata might talk a little bit about these technologies now, and then I'll come back to you towards the end.
Yeah, thank you, Will. So let's start with not so new technology that you have heard previously, as Emma mentioned, as part of the NHS, that sometimes whole genome sequencing is done. So since I am a bioinformatician, then I thought to tell you a little bit of what does it actually mean to sequence genomes? How is it done? So how does whole genome sequencing work? So whole genome sequencing simply means to read all of these letters in a person's DNA. So let's read all of these A's, C's, and G's and T's in a person's DNA, which is 3 billion letters, which is a lot. Now, unfortunately, we cannot read it, sequence it as a whole molecule. So the way technology works, we have to shred all these letters in smaller chunks, in smaller pieces. And then we have machines that can read each piece. And then we have to put all these pieces together in order to read that book, to read the sequence of the letters, yeah? It's very much like if you think about a big jigsaw puzzle, doing a very big jigsaw puzzle with very many small pieces, yeah? So I needed to shred my DNA. Then when I sequence it, I can read every piece, but then I have to paste my pieces together. So here I have this like blue picture, and if you do jigsaw puzzle, sometimes, you know, at the end, you're left with these blue pieces of sky or green pieces of grass, or I don't know, like in a corner, you're not quite sure In particular, if pieces are very small, you're not quite sure where it goes, yeah? So when we say reads for my DNA, it's like pieces of DNA that machine sequence, yeah? And the bigger the pieces, the easier it is for me to assemble it, and less likely I am to make an error when I put these pieces together, yeah? So if I sequenced a person with using whole genome sequencing and I didn't find a variant, a mutation, maybe I didn't find it because these small pieces, because the mutation was on that small piece of sky, let's say, and I wasn't sure where it belonged, so I don't see it clearly in my picture. And one thing to do is to try now to assemble the same picture, but using larger pieces, jigsaw puzzle with a smaller number of bigger pieces, yeah? So this is what we mean by long read sequencing. So this really very promising new technology, long read sequencing, it means that now we still need to shred the DNA in order to read the same 3 billion letters, but now we're shredding it in bigger pieces. And so long reads simply means bigger pieces. And now it's easier for me to assemble it. You see, like when you buy a jigsaw puzzle for a small child with less developed skills to assemble the puzzle, then you would go for puzzle that has like a smaller number of large pieces. So exactly like here, so if I go for this long read sequencing, one jigsaw piece is big, and when I put it together, it's easier for me. My picture is clearer, and I am much less likely to make mistake in assembling it. Now, we're still reading the same 3 billion letters, yeah? But our picture is genome wide. I see every position in genome now much more clearly. But particularly, I love this technology for mitochondrial DNA. As Emma has said, our DNA is big, but our mitochondrial DNA is small, and our mitochondrial DNA is around 60,000 of these, 16,000 of these letters, and that means that now one of my jigsaw pieces is big enough to include the entire mitochondrial molecule. So actually, if something has gone very wrong with mitochondrial DNA, I will now see it really very, very clearly. I almost don't need to assemble it because that's how clear it is. So it's a really very, very nice technology. That's long. So if you hear next time long read sequencing, that's what it means. The same sequencing, like whole genome sequencing, just I'm more confident because I had less work to do to assemble it, and my picture is now clearer. That's what it means. So let's say that I've done that. I also use better bioinformatics tool, as Will said before. We also always need to read about newer bioinformatics tools and to try them. And many times replacing a tool led to new diagnosis. So let's say I either use a better tool or bigger jigsaw piece. I use longer read sequencing, and I may have found a wrong letter in patient's DNA that we believe causes their symptoms. So you have heard from Charlotte and from Emma, how does it work that we need to have these points of evidence, and now I'm not actually sure. I found the wrong letter. I found a variant, but it's so rare. We never saw it before, and we really need to make sure that it's really the culprit. And how do we do that? So now I'm moving into another new technology here, yeah, that comes after DNA sequencing. So I'm sure that most of you have heard this analogy that DNA is like a recipe that makes us us, yeah? So let's look at DNA is our genome, and I'm looking at one recipe in that recipe book, and let's say that this recipe is for Granny's sugar cookies, yeah? And I see that in that recipe, one letter is a bit smudged, so this U, I don't think it's really a U. So this is, let's say that this is like the variant that I found, yeah? But I'm not sure, will I still get good cookies from this? So what can I do? So probably many of you have seen this as well. We used to call it central dogma of molecular biology, how you go from DNA to RNA and then to protein. So it's actually a little bit more complicated, but let's still use that, DNA, RNA, protein. So if DNA is my recipe, and the variant is that one wrong letter in my recipe book, then RNA would be like when I'm going to make these cookies. So RNA is like a handwritten note that the chef has scribbled. He copied it from the recipe book, and then he's taking it to the kitchen to make the cookies. Yeah. So my new technology, RNA-seq, is like, I know that there is a mistake in a recipe, like a one letter, but maybe it's going to be okay. Maybe he can work it out. But RNA-seq is like, I'm going to actually look at the scribbled note that he copied and is now making in a kitchen. And that can tell me something. So this is RNA-seq. So let's read what the chef has actually copied and tried to make. So again, Granny's sugar cookies. And I would hope that he will know that this is 2 cups of flour. Let's look at the handwritten note. And unfortunately, it says 2 cups of flour. So it did not work it out. That looks really wrong. So what he took to the kitchen really doesn't look great. And unfortunately, our protein that is supposed to be cookies is now I don't know whether he put like 2 cups of like something from the flour, or he stopped there, or he missed the flour, and it looks like he just baked butter. But now we have more evidence that this smudged letter, this U that was supposed to be that is not a U, and he put O there. Now we know that letter was actually very important, and we did not get a product that we needed to get. So even if you have done all of that, it can still take years and years to get a diagnosis. And how can we get doctors to think mito? So I'll pass it back to Will as a doctor who needs to think mito more than bioinformaticians do.
Thanks, Renata. Yeah, hopefully I am thinking mito if I work in a mito clinic. But I just wanted to touch on this. So this will be like our last slide, I think. And it's just saying that, I think it was really nicely put by Emma earlier that like before she gets the sample in her lab, some doctor has to go, there's something wrong with the muscle and the heart, this could be mitochondrial disease. But we know that not every doctor will ever have heard of mitochondrial disease, and most of them will never probably see a mitochondrial disease patient in their life. And, education can only go so far because, yes, we can try to educate a lot of neurologists and pediatricians, or metabolic doctors might be very good at this already, but not every doctor can be an expert on every rare disease because there's thousands and thousands of them. And so to get the diagnosis, we first have to recognize the pattern of symptoms to trigger the test. And so one thing that we are in early stages of looking at, but I think is exciting for the NHS in general, is taking AI and looking at the medical records of a patient, right? So you can look at, say, their letters, you could look at their MRI scans, their blood test reports, and then use AI to turn that into very standardized information. So if someone has, say, droopy eyelids, ptosis, that is written as ptosis for every patient, and that the age it comes on is known, etc. And so we get really nice descriptions of patients from AI pulling information from the patient notes, and we train the AI to do that really well. And then the next thing you do is use another AI tool to compare patients who are known to have mitochondrial disease and patients who don't have mitochondrial disease, and try to start to train an AI algorithm to be able to differentiate them. And the idea would be that in the future, we'd be able to build these alert systems. So say when, one of you guys rocked up to A&E when you were first having symptoms, the AI, machine or clinical decision aid tool would be able to say, okay, Mary has droopy eyelids, she's got a problem with her retina, and she's deaf, and this came on when she was 12. So the most likely thing is this is mitochondrial disease, and ideally something would ping up to the doctor and nurse and say, have you thought about mito? This is the doctor to write to, or this is the test to send to, Emma and Charlotte's lab is at this address, you'd send this kind of blood test, etc. So we'd really like to get that going. So, in our clinic, we're in the very early stages of starting to train this information on patients that we have consent to work on. But in the future, we'd really love to build this further. And the Lilly Foundation are setting up this mitochondrial research institute where they're going to have a big kind of registry of patients and also try to work with the NHS to make all these systems better. And of course, the more patients that we put into these things, the better, and the more standardized the data. So this is quite early, but I think it's an exciting time. And if we can raise enough money to do these studies really well, it could it could get the samples to Emma a lot earlier, get the diagnosis and get us in there seeing the patients, getting the screening going, and hopefully at some point when we have more clinical trials, getting them going earlier. So I think it's an exciting time to work in this. So I think that's our last slide, Renata. So we just we're happy to take any questions, and I'll let Katie take back over now, I think.
Thank you, Will. Thank you, Renata, Emma, and Charlotte. That was fantastic. Lots of information. I just want to say a massive thank you to the Newcastle team, the London team, for giving up the time and doing some fantastic presentations. So thank you all so much. And we will see you again all soon. Thank you.